Now we're going to look at nucleic acids, another group of large biological molecules that we also know are polymers made of monomers. Let's start with why they're important. Nucleic acids store and transmit hereditary information. Hereditary information is the information that can be passed from parent to offspring, and this can only be done with a type of molecule called DNA. DNA contains the code of life. The DNA sequence can be used to make genes, which are the units of inheritance that determine the traits that we inherit from our parents. For example, you have genes for eye color, skin color, hair color, height, etc. I'm sure you've heard of DNA. Do you know what it stands for? DNA stands for deoxyribonucleic acid. Have you ever heard of the other type of nucleic acid, or RNA, ribonucleic acid? These are the two types of nucleic acids. DNA is the molecule that does contain the genetic information, the raw information, but RNA is used to facilitate the process where DNA, in this case of the diagram shown here, can actually leave the nucleus of a cell and get to the ribosome. Part of the DNA sequence can actually be converted into messenger RNA that's then sent to the ribosome where the polypeptides are synthesized. And these ultimately will result in proteins, which is what controls the genes. So I just wanted to show you an overview of the importance of both DNA and RNA in the synthesis of a protein. And this is a process that we'll really study in more detail in a later chapter later this semester. Let's just try to focus on big picture stuff for now and go back to the chemistry aspect of things. Remember that nucleic acids are polymers, which are made of monomers, and the monomers of the monomers of nucleic acids, yes, are nucleotides. But each nucleotide is also made of specific components. Each nucleotide has three main components. One is a nitrogenous base. So nitrogenous bases would be, you've probably heard of the letters maybe, or you maybe even know the full name. For now, I'm just going to give you the letters. A for adenine, T for thymine, G for guanine, C for cytosine. Now, how these, the structures of these actually are either pyrimidines or purines. The pyrimidines have a 6-membered ring. The purines have a 6-membered ring fused to a 5-membered ring. Although this detail is on the slide, I won't be testing you in this chapter on that concept. That will come up later when we get more detail into the molecular genetics chapter. But I do want you to know the basic components of a nucleotide. So in addition to the nitrogenous base, there's going to be a pentose sugar. Pentose refers to the fact that it has 5 carbons in its structure. And there are 2 different types of pentose sugars that we see in the nucleic acids. Deoxyribose, which is in DNA, hence the name deoxyribonucleic acid. And in RNA, it's ribose, and hence the name ribonucleic acid comes from which of those pentose sugars are composing their structure. And thirdly, each nucleotide has a phosphate group. So these are necessary components for the nucleotides of both RNA and DNA. So this is something that they have in common, although you do see some differences there. Kind of zooming out a little bit to the bigger structure, DNA forms a double helix. There's going to be 2 strands of nucleotides that then wrap around each other and form a double helical structure. In contrast, RNA is a single strand, single polynucleotide. There's no double helix. There's really no regular shape it has. It actually can like kind of fold on itself and form other shapes within it, but it's a single strand versus DNA is always going to be a double strand. In DNA, I mentioned the nitrogenous bases adenine, thymine, guanine, and cytosine. You're always going to have this rule where adenine pairs with thymine, guanine pairs with cytosine. That's actually a rule we'll look at more in the future called Chargaff's rule. He was a biochemist who did some interesting research and determined that this is how they must pair together. And we can see that through the next slide. Come on, next slide. Here it is. Okay, so this is a DNA double helix that's actually undergoing DNA replication, where at the bottom, where the double helix parental strands in blue have kind of uncoiled, unraveled from each other, and now they're each serving as a template for a new strand of DNA to be formed, which are shown in orange. What I want you to look at here, just the complementary bases. Notice that wherever you have thymine, there's 3 black dots there. Those are hydrogen bonds linking those complementary bases together. So thymine is hydrogen bonded to adenine, cytosine to guanine. You can see that in the original parental strands and also where the parental strands have separated and are now forming, serving as templates for new daughter strands to form. So A and T, C and G. So I also wanted to emphasize here that hydrogen bonds join the base pairs together, but the backbone of DNA is actually held together by covalent bonds. And although those are not shown, it's a stronger bond, it's more permanent than the hydrogen bonds, which are temporary and can easily be broken and also reformed. And that's important because they need to be able to break and reform in order for DNA to replicate. Preparing for cell division, each of those DNA strands needs to serve as a template, so they have to be able to separate from each other to do this process. And then the result of that will then be 2 identical copies of the original double-stranded DNA molecule that will then end up in 2 daughter cells. So through the process of either mitosis or meiosis, we'll look at that later in the semester, and then the role of the DNA in that process. So again, we're just kind of skimming the surface of this concept of nucleic acids and understanding how these are important biological molecules that we'll study in more depth later in the semester.